Good morning. Good morning and welcome to this last session, the Closing Plenary session. I hope you enjoyed this meeting. And now it's time to move to basic science. So basic science fueling personalized medicine for cystic fibrosis. I have no conflict of interest. If we think to these words, basic science and personalized medicine, well, they are quite different. Basic science, or pure science, aims to advance fundamental knowledge regardless of immediate practical applications. And, well, personalized medicine is absolutely the most practical things that we can do for people with cystic fibrosis. Nevertheless, basic science played a fundamental role in advancing our knowledge on CF disease and also to develop treatments for this pathology. Here you see a scheme that summarizes the milestone in CF research, and in green you can see highlighted the steps that were brought to you by academic basic science research. This knowledge during these more than 30 years has ensured a better understanding of CFTR biology and CF physiopathology more in general, and had allowed to translate this basic science into changes in clinical practice. I would like to highlight some of these steps with you. If we think about rescuing the CF basic defect, everything, we can say that apart from the characterization of CFTR protein, its function and defect in CF, the first meaningful step was the design of novel assay, cell-based assay that allowed the high-throughput screening of large chemical libraries to identify modulators. These required the development of assays that were suitable for this large screening, and here you see an example. Probably the most used assay for high-throughput screening developed by Luis Galleta and Alan Workman. And it's based on a special variant of a yellow fluorescent protein. And thanks to this novel assay, it was possible to run the first high-throughput screening that identified the first CFTR potentiators, and in 2005, the very first corrector of F508del CFTR. This drug discovery project was coordinated by Alan Workman at the University of California, San Francisco, and led to the identification of 5 classes of correctors. Unfortunately, none of them was further developed or maybe was adequate to be developed. So we have no drugs derived from the very first screening, apart from the proof of concept that it was possible to rescue the maturation defect of F508del CFTR. In those years, other 2 projects, drug discovery projects, were ongoing. One at the McGill University in Canada, coordinated by John Harran. They identify an approved drug, sildenafil, and also some of his analogs as correctors of F508del. And then there was also, obviously, the high-throughput screening that led to the identification of the Vertex compounds, and we all know how this ended. A few years ago, scientists started to focus on unraveling CFTR structure, and they started to try to understand the structure of the nucleotide binding domain 1, where the F508del mutation is located. And they achieved to understand the structure of NBD1 in the normal, but also in the mutant protein. And this led to a new round of a drug discovery project. I want to mention the EPICS project, in which the scientists aim to identify, to design novel correctors based on the structure of the target protein. And indeed, they identify several compounds with different chemotypes having activity as not only as corrector, but also as potentiator, and even molecules having dual activity. This taught us that there is a profound, a deep link between CFTR structure and its activity. And then we suddenly realized that CFTR is not alone inside the cells. There are many proteins and molecules that can interact and somehow regulate CFTR biogenesis, maturation, trafficking to the membrane, and also activity. And this new knowledge brought also a different concept, the concept that we could try to target the interactome to rescue mutant CFTR. And this opened a completely new field of research that led to many different manuscript papers, many different studies, demonstrating that there are many proteins that can be targeted, and this leads to CFTR rescue. However, until now, there is no alternative target that we can safely modulate to rescue mutant CFTR. They could be particularly important not only for cystic fibrosis, but also for other diseases. However, the safety of this kind of maneuver could be not so clear. So, if we have to summarize what we can call an achievement of basic science in the very first years of research, well, if we look at the very first decade, it was fundamental to understand CFTR function and also to clarify CF basic defects, in particular related to the F508del mutation. And in the following 20 years, well, those were the years in which basic science could identify small molecule able to rescue F508del, but it was also the time for investigating CFTR structure and its link with channel function. And finally, we unraveled CFTR interactome and regulome, and this also led to the fundamental concept of the role of cellular environment and cellular background in CFTR biogenesis and maturation. 3 years ago, I had the honor to chair, together with two great scientists, Carlos Farina and Jeff Brodsky, the Basic Science Conference. And first of all, we should acknowledge the fundamental role of the ECFS in supporting basic science through letting us organize and supporting every year since 2004 the organization of this important meeting that is open not only to European scientists but also to overseas scientists. And when we had to write an editorial about that particular conference, Carlos Farina had the idea to entitle it Fundamental and Translational Research in Cystic Fibrosis, Why We Still Need It. And in this editorial, we highlighted the new challenges that were in front of us 3 years ago. So we know that there are still features of CFTR folding, of CFTR function, and also of its pharmacology that are not completely understood, and they will be fundamental to design better and better drugs, treatment for all the people with CF. We don't fully understand the physiological mechanism that regulates CFTR activity and expression in native tissues, not only in the lungs, but also in the other organs, where the role of CFTR has not been completely clarified. And there is also a lot to understand about CF lung environment, because mucus and the vicious cycle with infection and inflammation plays a fundamental role on defining the lung disease. So CFTR rescue could not be enough to revert completely the disease, and we are well aware of this. And finally, we still have to identify effective therapies for every individual with CF. That is a huge challenge. We still need to optimize our personalized medicine approaches for rare mutation, and this will require also the development of innovative gene-based approaches, because some of the mutations that are present in people with CF cannot be treated with modulators. And we also need to understand the mechanism of disease and also the mechanism of action of some of the therapies that we are using, because this will let us to identify novel drug targets to improve CFTR rescue and, more in general, the lung condition. To date, in Italy and in other European countries, particularly in the south of Europe, we have a consistent percentage of people with CF that are still orphan of a treatment, because they have a mutation different from F508del, for which CFTR modulating drugs have not been approved yet, or in some cases they have been approved, but they are not yet available for people with CF. And what was the work done by basic research in these years to meet this unmet need? Well, basic science played a fundamental role in generating new predictive patient-derived cell models to assess efficacy of available drugs on rare mutation. And this means that the first step was to clarify the mechanism by which these mutations were causing loss of function, and then to see whether it was possible to rescue the activity of these mutant proteins. Among the cell models that were developed, there are intestinal organoids and nasal epithelial cells. Intestinal organoids were, in absolute, the first predictive model developed in 2013. They are based on rectal biopsies. You can retrieve the cells, expand them, and then generate this organoid that can swell based on the amount of CFTR activity. And they are very useful to monitor and to investigate mutant CFTR activity in patient-derived samples, in particular for those with rare mutation, and to address their responsiveness to drugs. Repeatability and reproducibility for this kind of assay was consistently confirmed and validated by two of the most important European labs working with intestinal organoids, the one localized in Utrecht and the one in Leuven. And also it is feasible and safe to centralize the generation of these intestinal organoids to test them in, let's say, clinical trials. The second predictive models I want to highlight is nasal epithelial cells. The nasal epithelial cells can be obtained by nasal brushing. The cells can be expanded, differentiated in vitro, and used for direct measurement by electrophysiological techniques of CFTR and also other channel activity. And the laboratory of Isabelle Sermet did a fundamental role in optimizing the protocol for the culture of these cells, and also to demonstrate that it was possible to compare the activity and rescue of CFTR function in samples derived from patients with the activity, with the clinical benefit from people that were recruited in the frame of the compassionate use program in France. In this work, they clearly demonstrate that there is a correlation between CFTR rescue measured in nasal cells and changes in FEV1 and also in sweat chloride content. Our lab is also involved in this type of studies. We have a huge teratyping project running in our lab in Gaslini Institute. It was fundamental to establish a collaborative network with many CF centers in Italy, and we are now collaborating with 16 different CF centers. But a fundamental role was played by the one in Gaslini Institute, headed by Carlo Castellani, and also other centers were fundamental because of the high number of people that were recruited in the frame of the project, and I want to mention and thank Florence and Messina in particular. In the frame of this project, we recruited more than 450 donors. We have already completed the analysis for approximately 400 of these donors, and we sent out more than 350 reports to the clinician. Hopefully, these reports have been useful for them to see whether it was possible to treat even off-label these people, but also in some cases to clarify the diagnosis that had some uncertainty. Here in this graph, you see a summary of all the results that we obtain. Each dot is a sample derived from a different donor, and here you see in untreated condition, in most of the samples, the CFTR activity measure is extremely low, and here on the right there is the distribution of the CFTR activity measure in the same samples, but after treatment with the most effective modulators that were identified for each sample, each genotype. And you see that there is a huge increase. Indeed, as also demonstrated by other studies, what we observe is that many orphan, mainly missense mutation, can be rescued by modulators. We still have some variants, also missense variants, that are not rescued, and they will need optimized drugs. But this study also highlighted the importance of in-depth molecular analysis, because we observed something quite strange and disappointing at first. The Genoa CF Center identified 2 patients having one F508del allele in combination with a null allele, minimal function, not rescuable allele, that were clinical non-responder. And the nasal cells were very precious for these 2 people because we could obtain the cells and investigate CFTR activity and rescue in those cells, and we could confirm that the problem was within the cells and not related to, for example, drug metabolism. And an in-depth molecular investigation identified a 3rd substitution, the L467F, that was previously unidentified. This is not even a CF-causing mutation, but this mutation in these people was in cis, so on the same gene copy with the F508del. And this protein encoded by this double mutant allele is not responsive to ETI. A preliminary investigation in our genomic database confirmed that this complex allele could account for 3–4% of all F508del allele. And now, more recent studies have highlighted that this complex allele could reach frequency among F508del alleles up to 8%, for example, in the Russian Federation, but it is quite low in other populations. For example, in the US, it amounts for about 0.3% of all F508del alleles. So if we consider the number of non-responders that were highlighted also in the trial for ETI, I think that we could reasonably say that the majority, at least the majority of these non-responders are indeed people carrying F508del complex allele. And this is something that should be considered by basic science and also by pharma companies that are developing new drugs. And the sad news is that we are currently investigating 2 novel F508del complex alleles not reported in the literature that were identified in 4 novel non-responder, clinical non-responder people with CF. Another fundamental support of the European Cystic Fibrosis Society to basic science is the support of working groups, especially those, in this case, that aim to develop or optimize these predictive models. And I want to mention the airway epithelial cell model for diagnostics. It is coordinated by Isabelle Sermet and myself, and the first achievement, the first result of this working group was the generation of standard operating procedures for cell culture, differentiation, and evaluation. These have been done in collaboration with many other renowned laboratories that use this type of cells in Europe, and it has been also shared with the main laboratories working with these cells overseas. The training for more, let's say, newer participating labs is ongoing this year. And I want also to highlight a new working group coordinated by Jeff Bickman that will do probably something similar on intestinal organoids. So we know that EMA has approved finally ETI also for a consistent fraction of these 30% of people that is still orphan of a treatment. And in the approval of this extension, it is mentioned that the ETI will be usable for all people with CF, provided that they do not harbor 2 class 1 mutations. But what is a class 1 mutation? Are we sure that we know what a class 1 mutation is? Here you see one of the most used schemes representing the classification of CFTR mutation into the 6 or 7 classes. Class 1 says that the defect type is no protein, and we have 3 mutation examples. All of them are PTC mutation. So nonsense mutation introducing a premature termination codon in the sequence. For this reason, I probably prefer, and I want to highlight this other scheme. It also reports the classification in different classes. But here, in the type of mutation, you see that there are mentioned not only nonsense mutation, but also frame shift and canonical splice site mutation. I think this is more understandable, not only to basic science, but in particular to people involved in the clinical settings. So what about this canonical splice site mutation? These mutations are usually grouped into class 1, and these are mutations occurring in a very conserved region in the RNA, and this region marks the boundary between an exon and an intron. So it is fundamental for the cut and paste process that leads to a mature messenger RNA that can be translated into a protein. Well, if we go back to the scheme, we will see that here, the canonical splice site, for example, the position +1, when you see a +1 after the number of the nucleotide where the mutation is, it's indeed in class 1, but surprise, there is also a canonical splice site mutation that belongs to a class 5. So it is not so immediate to establish whether even a mutation in the canonical splice site is a class 1. So a canonical splice site mutation is not necessarily a class 1 mutation. It is rare, a rare event, but sometimes it is not a class 1 mutation. What about nonsense mutation? They should be grouped in class 1. These are mutations that change the sequence by introducing a stop codon where there should have been an amino acid coding codon. And also in this case, basic science could identify nonsense mutations such as this EA31X, in which the mutation causes a different defect. It causes an aberrant splicing, so you have some transcripts that bear the PTC, but also you have some transcripts in which you have the deletion of this amino acid, and this is a protein that retains a wild-type activity. So also in the case of nonsense mutation, it is not necessarily, nonsense mutation is not necessarily a class 1 mutation. And what about deletion, in particular large deletion, or deletion that involve also rearrangement? Well, they usually involve also the skipping of multiple exons, and they are usually associated with a more severe phenotype. Well, they should be class 1, but also in this case there could be some surprise. These are two examples, two samples from two people having exactly the same large deletion that led to a rearrangement. And in both these patients, we observed the cells from this patient, we observed some CFTR activity that could be increased upon treatment with ETI. There was a third person bearing the same mutation. We saw some effect, but we could not reach statistical significance for this result. And we could demonstrate that also in this case, the reason for this activity and response to ETI resides in an aberrant splicing that generates an in-frame transcript with the skipping, an in-frame skipping of an exon and a residual activity and response to ETI. So even a deletion or even a large deletion is not necessarily a class 1 mutation. So classification of mutation into class 1 should be done after careful evaluation of their effects at the molecular and functional level, and this could be one of the new role of basic science. And for this reason, I hope that you understand that we still need fundamental and translational research in cystic fibrosis to understand why. We could do, as Pierre-Régis suggested in his opening lecture, to give these people the drug and to see whether they respond. But I think it is also important to understand, in this case, why they respond. And thank you very much for your attention.
Thank you very much, Nicoletta, for an excellent and very clear explanation of some quite complex stuff. No, not yet. But also for all of the work that you've done with your co-presidents and vice presidents for the conference. We very much appreciate it. And you're quite right when you highlight the importance that ECFS in general puts on basic science and all the work that you and your colleagues are doing.